Hello, dears, and welcome to Al-Hussaini Virtual Lab Pathology Talks, Tips, and Practical Tips. What I'm going to be sharing with you today is a nice example of a rare and recently described tumor, which is the CNS CIC-rearranged Sarcoma. So this is a case of a 14-year-old male patient who was diagnosed a few years back as ALL, for which he was treated and cured. He presented with a large right frontal lobe mass that was completely excised. And as you can see here from the low power magnification, the tumor is kind of pink rather than blue in color. And this is important actually whenever we render the differential diagnosis. There is this tumor growing in sheets with no particular growth pattern. On higher power magnification, and again, as you can see, the tumor cells have abundant acidophilic cytoplasm. They have this very peculiar vesicular nucleus and prominent nucleolus. On other areas, the tumor cells show this vesicular or vacuolated actually cytoplasm, still showing the vesicular nuclei and prominent nucleoli, and the scattered mitotic figures were encountered among the tumor cells. Other areas showed this myxoidish background, and in some areas there were a tendency to spindling of the tumor cells, not very prominent actually, but it was very focally identified. Now, in other areas, and to me, it looked like it was resembling a typical teratoid rhabdoid tumor. Actually, this was my primary differential diagnosis of this unusually brain tumor. We have the cells with abundant acidophilic cytoplasm. We have this vesicular nucleus and the prominent nucleolus, and then we have this myxoidish background that became even clear more and more, the vacuolated background, the cells with the prominent nucleolus and the vesicular nucleus, and the cells which some of them have abundant acidophilic cytoplasm. Again, here it was very much reminiscent of a typical teratoid rhabdoid tumor cells. Some areas, here's other tumor cell areas, again showing these cells with abundant acidophilic cytoplasm, this billy-like cytoplasm containing filaments, and the nuclei, vesicular nuclei with the prominent nucleoli, and then the myxoidish background. Again, look here at the prominent nucleolus. Honestly, to me, the first differential diagnosis was a typical teratoid rhabdoid tumor. And again, another focus where we can see the appearance of the tumor cells. This is just to give you an idea and a depiction of how sometimes the tumors can really look alike. And we have to consider why differential diagnosis whenever we encountered unusual tumors. Now, the tumor was diffusely positive with CD99. And this, again, it triggered another differential diagnosis, Ewing sarcoma. But the NKX2.2 immunostain was completely negative, which rules out essentially the diagnosis of Ewing sarcoma. Now, WT1 was done at a later stage, and the tumor cell nuclei were positive with WT1 in support of the unusual CIC-rearranged sarcoma. Synaptophysin showed only very focal scattered positive tumor cells. Because of the differential diagnosis as rhabdoid tumor at the first instance, I did the INI1 or the SMART-B1 immunohistochemistry, and there is retention of the nuclear stain, and the P53 only showed the mosaic growth staining pattern consistent with wild type staining pattern. At this stage, actually, the tumor was diagnosed as malignant tumor, and the tumor was subject to methylation profiling, and the methylation classification came back as tumors of uncertain differentiation consistent with CIC-rearranged sarcoma with a calibrated score of 0.99, which means a confident diagnosis. So we went back and did FISH for fluorescence in situ hybridization to look for the rearrangement involving the CIC CIC gene on chromosome number 19, and this is a break apart a probe where you see normally the yellow colored dot, which means that there is overlying signal. What we look for in order to render the diagnosis or the presence of the break apart is the separate green and red signals in the presence of one yellow signal per nucleus. We have to see this appearance in more than 10% of the tumor cell nuclei under fluorescence microscopy in order to render a confident diagnosis. Again, this is a beautiful example where we have the green and the red signal separate, which means that we have the CIC rearrangement consistent or supporting of the diagnosis of CIC-rearranged sarcoma in this case. So the final diagnosis that was rendered for this unfortunate child is CNS CIC-rearranged sarcoma. The patient underwent extensive imaging, including PET scan, and there was no evidence of CIC sarcoma anywhere else in the body. So this is consistent with the primary CNS CIC-rearranged sarcoma. Remember that this is among the rare tumors that were recently described, best classified by methylation profiling, but also can be identified by various other methods, including immunohistochemistry, where we find some positivity with CD99. It can be focal, it can be more diffuse, like in our case, doubled with WT1 immunohistochemical positivity, and in places where they have the ETV4 immunohistochemistry, it's usually positive. We don't have the stain. Other modalities to confirm the diagnosis is by fluorescence in situ hybridization or FISH, looking for the break apart of the signal in more than 10% of the tumor cell nuclei, as in this case, or with more advanced technologies like the methylation profiling or the NGS testing for the presence or absence of that translocation. Remember that the tumor is very aggressive type of sarcoma, primarily occurs in the soft tissue. 10% of cases occur in the viscera, including the central nervous system as a primary site. But whenever we encounter such a tumor in the central nervous system, we have to rule out metastasis. I hope you find this tip useful in your daily practice. Thank you.